Okay, let's look into pollination and fertilization. So let's define pollination. By the word itself, you know that it has something to do with the pollen grains. Okay, and you have to mention that it is a process. So what happened to the pollen grains? It is being transferred. So from where to where? It's from the anther. Remember, that's the stamen, that's the male reproductive parts, the place where pollen grains are being produced. So it's being transferred from the anther to the stigma. Okay, so stigma is the female reproductive parts, the couple. It's a place where it is to receive the pollen grains. So this process is known as pollination. So from here, pollination, we have pollinating agents. Okay, so that means they are the agents that is responsible to do this transferring of the pollen grains. So who are they? They are the insects, okay, like the bees, okay, like the butterflies, other insects, mammals, including human beings, sometimes purposely, okay, sometimes unintentionally. We also have the birds as well as the non-living things, water, and also wind can also serve to pollinate, to help in the process of pollination. So the presence of pollen grains on the stigma, that means if the stigma now they are having pollen grains on it, what's going to happen next is that it will trigger or it will stimulate another process which is known as the fertilization. Okay, and we know that in human beings also we have fertilization when the zygote is being formed from the fusion of sperm and ovum. But in the flowering plants, uh, yes, fertilization is present. There is this process, and it is the fusion between the male gametes and the female gametes. Okay, now let's look at the formation of pollen tubes and also the male gametes. Okay, now for this, you have to recall the previous lesson, okay, where we talk about the formation of the pollen grains. Okay, we end up having this pollen grain, the mature pollen, consisting of these two nucleus, which is the generative nucleus and also the tube nucleus. So what happened next? Okay, now we start our story here, number one, by saying that the wall of the anther, now this is the anther of a mature pollen. Okay, what will happen is that this wall is going to split. Okay, it will dry up, it will shrink, and then it is splitting, and therefore releasing all these pollen grains from the sac, from the pollen sac. So the released pollen grains, what happened? They will be transferred to the stigma. Now this is the female parts. So from here, enter going to the stigma. So this is what happened. They are transferred to the stigma. So this is the process of pollination. It might be the stigma of the same flower or different flowers. Okay, and this process is being assisted by those pollinating agents that we have mentioned just now. So pollen grains that have been transferred to the stigma will germinate. So when they sit on these, okay, the sugary substance, yeah, so it's going to germinate. These pollen grains is going to germinate, which means they will form a pollen tube. Okay, pollen tube. So this is the pollen tube. Okay, pollen grain, and then pollen tube is being formed. So that means this comes from the tube nucleus over here. So pollen tube grows down towards the ovule. So it's going down all the way down into the ovule. Okay, and it is through the styles. Remember the style, this is it, parts of the carpel. So the generative nucleus, okay, now we have two, right? Remember this, tube nucleus and tube nucleus over here, and also generative nucleus. So just now, formation of pollen tube. Then now we talk about the generative nucleus, which will move along the pollen tube towards the ovule just now. At the same time, the generative nucleus will divide. Now, where is the generative nucleus? Okay, so it's moving along this pollen tube. It's going to divide by means of mitosis. That means from one nucleus, we'll end up having two gametes, two nucleus. That is mitosis, from 1 to 2. Okay, next is that the end of the pollen tube will secrete an enzyme. The end of the pollen tube, there will be enzyme secretor and it's going to digest the tissues of the style. So when it reaches the embryo sac, okay, this is the embryo sac housing the ovule. So the pollen tube will penetrate the ovule through the opening. This is the micropyle. Pollen tube is going to penetrate into the ovule through the micropyle, where the tube nucleus will degenerate. So at the end here, not forget that this tube nucleus is here leading. When it first reaches this point, it's going to degenerate. When it degenerates, can you imagine that it's going to leave a hole over here, and therefore it's like a door, serving a door to open up for these two male gametes to enter into the embryo sac. Okay, so that's what happened. So next is that now we talk about fertilization, double fertilization. Double, by the terms itself, double, two, process of fertilization in the formation of diploid zygote from the first male gamete, and then there's another triploid nucleus. So double fertilization involves two male gametes, which the first one will fertilize the egg cell, forming diploid zygote. The second male gamete is going to fertilize or to fuse with the polar nuclei, forming a triploid endosperm tissue. Okay, remember that we have mentioned generative nucleus is going to undergo mitosis, right, in the pollen tube, and therefore that's how you get the two male gametes. Okay, and then these two male gametes are haploid, and then they are entering the embryo sac. Inside there, what happened? Fertilization. So let's look at the picture here. So this is inside the ovule. Now you can get to see this 1, 2, 3, 4, 5, 6, 7, 8. Still remember our 8 nuclei the other day when we discussed about the formation of the pollen grain. Okay, 3 rounds, the microspore cells undergo 3 rounds of mitosis, and therefore that's how they end up getting 8 nuclei. So they are now positioning themselves accordingly. 3 here on one pole, that's the antipodal cells. Okay, another 3 here, okay, with 1 egg cell and then 2 synergid cells in between. Okay, the egg cells is in between these 2 synergid cells, and we have the 2 polar nuclei, which is in the middle. Okay, the other end we have the 3 antipodal cells. So these middle 2 polar nuclei, they are without the membrane plasma. Okay, so the egg cell, okay, which is the most important one, okay, is having these 2 synergid cells on the left and at the right. So what happens? So this is the first step. Upon reaching the embryo sac, this pollen tube, just now already mentioned, it will penetrate into the ovule. This is the ovule through the micropyle, and that's the opening over here. So the tube nucleus will degenerate. So the black one, this is the tube nucleus. It is going to degenerate, meaning there will be a hole here. There will be a door, an opening to let these 2 male gametes enter into the embryo sac. Okay, the 2 male gametes to enter. So next, we have 2 male gametes now. So the first one, is going to fertilize the egg. Now in the middle here, it's going to fertilize it. Egg cells and also the male gametes, they are N, they are haploid. So upon fusing, what will be the products? It will be a zygote, which is diploid 2N. Okay, there's another male gamete. Do not forget that it will fuse with the two polar nuclei here in the middle. 2 plus 1. So that's how you get 3. Triploid endosperm nucleus. Okay, so this endosperm nucleus, later on we'll discuss the significance of this tissue. So that's the fate of these two male gametes upon entering the embryo sac. Okay, now the importance of double fertilization for the survival. So you see, fusion of one of the male gametes with the egg cells, okay, it's going to produce zygote, which is diploid. So that's why here we have the first statement saying the genetic information is passed down from one generation to the next. Okay, when zygote is being formed. Okay, and then this also will result in restoring the haploid condition in the gametes with the formations of diploid zygote. Okay, the haploid condition. Because we have these haploid gametes, therefore, when the gametes fused, the male and the female gametes fused, therefore, we can get a diploid zygote. Okay, that's the meaning of this restoring haploid condition in gametes. Okay, number 2, the second significance is that the fusion of another male gamete with the two polar nuclei, 2 plus 1 equals 3, and that is the triploid endosperm tissue. So what is so significant or important about this tissue is that it is being used for development of an embryo for the survival of the plant species. Okay, so in eudicots, okay, examples given over here, what happened is that they are making fully used of this endosperm for the embryo to develop before the seed matures. And in most monocots, okay, examples, coconut, wheat, barley, corn, only a part of the endosperm is utilized. Only a part is being utilized for the development of embryo. Some of them are stored in the cotyledon to be utilized during germination of seed, okay, which the endosperm tissues will enable the embryo to survive in the seed for a long time. Okay, if the conditions are not favorable for germination to happen, what happens is that this embryo will not die, but then it will still survive. How? By depending on the nutrients from these endosperm tissues. Okay, so these are the importance of double fertilization for the survival of flowering plants. Now let's go into this activity 6.4. The aim is to carry out activity to observe germination of pollen grains, formation of pollen tubes in sugar solutions under light microscopes. So we are using the cavity glass slide. Okay, this cover slips, forceps, mounting needle, droppers, and also light microscopes. So these are the procedures. So now let's look at the questions over here. First is that why there is a need to use fresh flowers. Okay, because we want to obtain fresh pollen grains that are able to germinate. Okay, and then describe the specific shape, characteristics of the pollen grain under the light microscope. Now this varies depending on the student's specimen, the student's observation. Okay, and then number 3 is that there is this group of students, they did not follow the correct procedure. Okay, suppose they use 10% of sucrose solution here, but that they make use of distilled water. So what will be the outcome? Now you have to think, why is this sucrose solution significant? Because we want germination to happen. For germination to happen, it requires energy. So this energy is supplied by the sucrose. Okay, it is supplied by sucrose, and therefore without sucrose, formation of pollen grains do not happen. Okay, because germination requires energy. Okay, then number 4 here is that there is this stain solution, acetocarmine solution. Okay, if they are not staining the specimen with this solution, what will be the problems? So there is significant in terms of the materials being used. We have to use this acetocarmine solution. The pollen tube will be difficult to observe. Why? Because it is colorless. So that means we have to stain it with this acetocarmine stain solution because we want to get a good view of the pollen tube. Okay, that's it for this 6.3.